Thank you for having me here, and thank you, Dr. Chen, for this warm introduction. So I'm going to share my screen, and we get started. Okay. Cool. Okay. So I hope everyone can see the screen well. So the title of my talk today is Spatial Atlas of the Mouse Central Nervous System at Molecular Resolution. This study is a joint efforts between the Dr. Xiao Wang's lab at Broad and Dr. Jia Liu's lab at Harvard. My co-authors on this work is a graduate student, Yichun He from Liu Lab, who is really the driving force for the computation part of this study. And also Dr. Yiming Zhou from our lab, who really helped with every aspect of handling the animal and annotating this huge data set. Okay, so the motivation of this study is really about the importance of studying cells at single cell resolution when we go to study tissues. I think for this audience, you probably already heard about this fruit smoothie analogy, where the bulk sequencing give you average measurement of a tissue sample. Single cell RNA sequencing can tell you what are the type of fruits and how and the abundance of individual type of fruit there in this mixture. So this gives birth to cell atlases that chart cell type diversities in the tissue and the organism, revolutionizing our understanding of the complexity of tissue composition and heterogeneity. And starting with a cell by gene expression matrix that characterizes individual cells in the high-dimensional gene expression space. And after dimensionality reduction, we can visualize the gene expression variation among cells, such as in this UMAP plot. Then by clustering cells and understand the features of gene expression in each of the cluster, we can then annotate what are the cell types and understand the potential functions they have in a tissue setting. So I don't have to emphasize how single-cell RNA sequencing has revolutionized our understanding of cell type complexity in the brain. Representative large-scale mouse brain datasets from Lindenson Lab, Allen Brain Institute, and the Dropvis dataset from Costco and Macaro labs have revealed, and many others have revealed, a few thousand of cell types in the adult mouse brain. Especially the diversity of the cells come from neuronal cell types that are mainly characterized by neurotransmitters. Besides neurons, there are also diverse glial cell types that perform supportive and protective roles, like immune cells, vascular, and epithelial cell types. However, a functional tissue is not just its cell type parts, but also the spatial arrangements and interactions among those cell types. That's why spatially resolved transcriptomics methods comes into play and recently named as the method of the year 2021 by Nature Methods. It bridges gene expression studies with tissue histology and connects gene expression space with the tissue architecture. For example, for a comprehensive understanding of brain physiology and especially brain disorders, it is critical to understand what are the gene expression features that is linked to a functional or pathological cell states. How do surrounding cell types respond to these disease-related cells? And if there's any regional specificity or regional specific vulnerability to such pathological states. Answering these questions require a single cell and a spatially resolved method, and ideally have a brain-wide coverage to give a comprehensive characterization. So the method we use in the lab is called STARMAP, Spatially Resolved Transcript Amplicon Readout Mapping. This is a method that developed by my postdoc advisor, Dr. Xiao Wang, during her postdoc time, and we continue to develop this method and platform in our lab. So this method combines in situ hybridization, tissue hydrogel chemistry, and in situ sequencing to really convert individual RNA molecules in a tissue sample into a sequence, into a punctal signal that can be sequenced under microscope. So to do so, we designed a pair of probes we call snail probes, composed of a primer and a padlock probe that anneal to adjacent sequences of a target RNA. And only when two probes both anneal correctly, the primer can serve as a template for DNA template DNA ligation to circularize the padlock probe nearby. And then the primer can initiate a rolling circle amplification to generate what we call a DNA amplicon. And there is also a red region on the padlock probe contain a gene unique identifier. And during this amplification process, we get thousands of copies of this identifier generated in situ. There is also modifications on the base we introduced during this amplification that can be further modified to be polarizable with acrylamide and bisacrylamide into a tissue hydrogel. After this, we can perform tissue clearing to further reduce background signal. And also we have this mechanically very stable sample for many rounds of reaction and imaging to get the barcode identity and the mRNA identity. So in this tissue amplicon hybrid hydrogel, we perform sequencing by ligation-based sequencing to get the identified sequence. So these two probe design, the Snell probe design, really ensures the mRNA detection efficiency, meaning that if there miss any of the probes here, the AND gate logic won't work, and you will see no signal from the sample. And the sequencing by ligation step, the CDL sequencing, can decode up to over 1,000 different genes in 6 rounds of imaging, where we add in a different sequence of the reading probe to decode the next 2 bases, depending on the specific sequence of the reading probe. Okay, so this method is, in summary, a targeted in situ RNA sequencing method that can work for biological tissue samples, has a single molecule sensitivity and a code mapping capacity of over 1,000 in the same sample. And combining tissue hydrogel chemistry and the tissue clearing, we can have really high signal to noise ratio in the 3D sample imaging. And when applied to a sample like the mouse primary visual cortex, we can image the gene expression of individual cells while retaining the physical coordinates of individual cells. That can give rise to this single cell gene expression matrix with spatial coordinates that after perform cell typing, we can achieve the spatial cell typing of a tissue sample. So for this study, we applied a StarMap Plus, which is an enhanced version of StarMap with improved probe specificity design. And we mapped over 1,022 genes in both coronal and sagittal slices from adult mouse brain. The targeted mRNAs were mapped at molecular resolution, and by segment those individual RNA dots into individual cells that we can have cell by gene matrix with the spatial coordinates for individual reads and cells. Just to give you a sense of how the raw data look like, here is a full hemisphere mouse brain slice in the first round of imaging, and we can zoom in into one of the imaging tile and further zoom in on a few cells. Individual dots in those images represent an amplicon generated from a single RNA molecule, and they appear as in different colors across these 6 imaging cycles. And this color array translates into mRNA gene identity. So in total, we collected a data set of over 1 million cells with 1,022 gene complexity with spatial coordinates. A little bit about the cell segmentation part, how we call out each individual single cells from those data set. The method we use is called StarMap, a cluster map, which is also developed in in the lab. The idea behind this algorithm is that by looking at the RNA dot image, there is a higher density of the dots surrounding the soma of individual cell. So we can call the boundary of a cell depending on the local dot density. We also take in the consideration of the dot, the mRNA identity into this segmentation. For example, when there are two distinct cell types nailing each other, we're able to draw the boundary of the cells depending on which cell type has the expression of certain which marker gene. Also, the StarMap Plus is a targeted sequencing approach. We were able to expand the gene list and predict transcriptome-wide spatial gene expression in single cells. This is done by integrating with paired single-cell RNA-seq dataset and gene imputation. Briefly, in this joint embedding space of the two datasets, the StarMap Plus cells, the spatially mapped cells, its transcriptome-wide expression is imputed as a weighted average of the nearest neighbors from the single-cell RNA-seq dataset. As a quality control, you are able to compare the imputed results with the StarMap Plus measurements we had, and also the in situ hybridization dataset from the Allen Institute. And both the regional markers and the cell type markers show these nice agreements among the three type of plots. And more encouragingly, for many genes that were not measured by StarMap, we observe consistent spatial patterns with corresponding images from the Allen Institute mouse brain in situ hybridization database, confirming the imputation performance. So not only cell type markers were well imputed, such as the GLUL, the marker for oligodendrocytes, or the PDE10A, a marker for striatal medium spinal neurons. When a gene is expressed in multiple regions, our imputation results can also predict the correct regional pattern and also the relative expression level across those multiple positive regions. For example, this NOVA1 nervous system specific splicing factor, and also this gene CTGF, which is positive both in the deeper cortical layer 6B, and also positive for blood vessel structures. So what can we learn from the spatially resolved single cell gene expression? We next aim to systematically understand the brain cell type diversity in the molecular expression and spatial dimensions. And here I will introduce the analysis framework we have. So in this analysis framework, we first identified molecular cell types by integrating with single cell gene expression dataset and validate with the nomenclature from orthogonal datasets such as Allen Institute. Then we were able to plot those cell types back into its spatial coordinates to get spatial cell type maps. The other aspect to fully utilize the single cell spatial information, this analysis define molecularly. So we cluster the molecularly defined tissue regions from the gene expression of the cell and its physical neighbors. And now we can compare this molecularly defined tissue region with the anatomically defined tissue region that can be derived by registering the brain slices into the common coordinate framework of the Allen Institute. So we can compare the differences and the similarities of molecular defined tissue architecture among different anatomical regions. And finally, by a combination definition will give us a comprehensive nomenclature system for individual cells containing both cell type and tissue region types defined by gene expression. In the following slides, I will go through more details of the analysis framework and more importantly, highlight some biological insights enabled by such mapping of single-cell gene expression in the context of brain tissue anatomy. The first part is about cell typing. We perform the two-layer cell typing at the mean cell type level. We integrate with a single-cell RNA-seq dataset from the Linderson lab and perform label transfer to align with the published cell type nomenclature. And we get 26 different main cell types. And then for individual main cell types, we perform the further laden clustering, resulting in 230 subtypes. And here's an example of the cerebral cortical inhibitory interneurons. We were able to identify all major categories of interneurons with canonical markers such as VIP, SST, and LAMP5, and a finer diversity within each category. And eventually it resulted in this large UMAP plot where the cells are colored by its subtype identity. So in total, we have, as I mentioned, 230 different subclusters. And indeed, the majority of the diversity comes from neuronal cell types. We have 190 neuronal types, plus a few dozen of glia cell types. Okay, and then finally we will have these spatial cell typing plots of all those over 1 million cells colored by its subtype identity. From the visually looking at these plots, we can already know there are clear molecular foundation that delineate the main brain structures, including the olfactory bulb, cerebral cortex, striatum, brainstem, and cerebellum. Really highlight the molecular foundation underlying the brain anatomy. And I would like to zoom in on this hippocampal region to just showcase the high spatial resolution we have in this data set. So in this plot, individual dots represent a RNA spot. And by drawing the boundary of the cells, we can also have an estimation of the soma size and the physical neighbors of individual cells. Here we'll check a few spatial plots of the main cell types to see what we can learn from these molecular cell type maps. First are the examples from excitatory and inhibitory neurons in the telecephalon, including the cerebral cortex and striatum. We see clear regionalization for most of the excitatory neuronal cell types, each occupying a defined brain structure or a defined cortical layer, while the inhibitory neurons tend to distribute more spread. Beyond the cortex, we also map spatial, we also have the spatial cell type maps for neuronal cell types in the thalamus, hypothalamus, and hindbrain. Especially, I want to highlight this rare cell type, HDC positive histologic neuronal cell type. Although it only accounts for about 0.1% of the cell population in our data set, we were able to forcefully identify them in the expected region, that's ventral media hypothalamus, with the correct marker expression. So that really showcase the power of spatial cell typing using our platform. Beyond neurons, our brain-wide spatial mapping also allow us to uncover the regional specificity of a few glia cell types. And I want to emphasize this is only achievable when we extend the mapping capacity to a brain-wide spatial mapping to really recover this large-scale spatial pattern of glia cell types. For example, the astrocytes. There are clearly different population of astrocytes in the telecephalon and the non-telecephalon region of the mouse brain. Another example is from the oligodendrocyte lineage, where more mature oligodendrocytes that marked by the orange color are enriched in the posterior parts of the brain, including pons and the medulla. This discovery highlights the importance of brain-wide mapping for uncovering such patterns and advancing our understanding of brain physiology and pathology. Okay, the second part of analysis is about these molecularly defined tissue regions. So we build molecularly defined tissue region maps directly from a term we call spatial niche gene expression. So we concatenated the single cell gene expression vector of a center cell with the surrounding cells ordered in ordered by the distance they have from the center cell. So by clustering the cells using this huge concatenated single cell gene expression vector and matrix, we're able to define the tissue regions by its molecular spatial gene expression. And as mentioned, we can compare this molecular definition with the anatomical definition after registering our sample into the common coordinate framework. And we were able to plot our molecular tissue regions in these three-dimensional mouse brain models and annotate each cluster with corresponding anatomical definition. Overall, the molecular defined tissue regions aligned well with the anatomical defined tissue regions. For instance, we observed laminar structures reflecting the layers of the cerebral cortex. And also there are tissue clusters reflecting different nuclei in the amygdala, thalamus, hypothalamus. So we spent a lot of efforts to do this annotation of individual tissue region clusters with the symbols describing the fine anatomical definition, the preferential distribution along the body axis, or dominant gene expression markers, following the anatomical nomenclature in the Allen Institute Mouse Brain Atlas. So these efforts really try to bridge the molecular expression space with the anatomical space to make those clusters more interpretable for neuroscientist biologists. With these two sets of molecular definition, we have this heat map plot to show the distribution of individual cell types across molecular tissue regions. And clearly, we can see the difference in the distribution or the regionalization extent comparing the neurons and non-neurons. The non-neurons are more widely distributed and have a larger scale spatial pattern compared to neurons. Within the neurons, we observe that many excitatory neurons reside in defined molecular tissue regions, as highlighted by this cross-diagonal pattern in the heat map. Resembling the patterns in the spatial maps we observed a few slides ago. So besides studying these cell types in the context of tissue regions, we also found many interesting observations by studying the molecular tissue regions and the gene expression features in the context of brain anatomy. So I'll continue with a few examples where the molecular tissue region can provide refinements into anatomical atlas. So for example, we found this finer laminar structure in the cerebral cortex layer 2-3, and with the paired supported by the marker gene spatial distribution. Another example is about the anatomical definition of the retrospinal cortex. We found there are different molecular tissue region definition for the anterior and posterior retrospinal cortex. In the anterior part of the brain, there are 3 molecular tissue regions corresponding to the retrospinal cortex. However, moving to the posterior part, this is not the case. And very interestingly, the posterior retrospinal cortex actually share similar molecular tissue region arrangements with the nearby visual cortex. So our study can provide insights into recent related study which identify that this posterior retrospinal cortex specifically has function in memory formation that involved visual function in rodents. So we propose that the molecular tissue region may be a more accurate aspect in delineating the retrospinal cortex definition and its subregions compared to the definition solely based on cell and tissue morphology. Another example is about the molecular gradients we can observe by looking at spatial gene expression patterns with single cell resolution. So this example is from the different lobules in the cerebellum cortex, where we see this transition between the dorsal and the ventral lobules of the cerebellum. And when extending this plot, we can observe when we plot the diffusion map component on the spatial plot, we see this gradient in different lobules. And this is also in agreement with a recent single-cell RNA study in the granule cells of the cerebellum, where a continuity rather than distinct cell types is observed in those granule cells. A final example about the molecular tissue region is this example from the dentate gyrus granule cells. So we found that a joint single cell and spatial definition of the cell type can resolve cell type heterogeneity better than the single cell gene expression itself. So when look at the molecular cell type definition, that we cluster solely by the single cell gene expression, these granule cells in the dentate gyrus largely form a homogeneous cluster. However, when we look at them in the spatial niche gene expression embedding, they clearly fall apart into two clusters corresponding to the dorsal and the ventral part of the hypothalamus, of the hippocampus. We're able to find the gene expression markers for this dorsal ventral transition. And it is also provide insights into lesion studies that reveal the dorsal and the ventral hippocampus have different functions. So this is a nice example highlighting spatial single cell mapping can really resolve the cell type heterogeneity related to function diversity. A second part about our study is the accessibility of gene manipulation. Why this is part important? So besides understand the cell type composition and arrangements in the tissue, more important aspects is to understand its function. And for drawing a conclusion about the cell function, there needs to be a way to manipulate the cell activity. For example, in the field of optogenetics, these light responsive channel rhodopsin proteins will be expressed in certain cell type to activate or deactivate the cell activity, and then we can track what's the functional outcome. For expressing those transgene molecular tools in neuronal cell types, a viral tool people use a lot is the recombinant adeno-associated virus. And a nice aspect about this virus is that depending on the specific capsid protein, the surface protein of the virus, and the interaction with the surface protein from the cells, there can be preferential uptake and expression of the virus in different cell types and the tissue region. And indeed, there are many engineering efforts surrounding the capsid protein of the AAV to achieve different organ and cell type preference. One example is from the Guadagnino lab and also the Derriman lab at the Broad Institute. They have developed this engineered variant called PHP-EB that's capable of penetrate the blood-brain barrier and achieve central nervous system-wide expression with a non-invasive systemic delivery through blood vessel. However, to which cell types these viruses express and quantitatively how well they express in individual cell types still need to be comprehensively characterized. So we incorporate this part of measurements into our data set, hoping to provide a chart and a resource for the community to help their choice of viral tools in the future. So for this purpose, we labeled the AAV genome with a sequence encoding a circular RNA barcodes under a generic pulse rheosix promoter. The barcodes were designed in a circular format, so there will be sufficient accumulation of the barcode copy numbers to allow a larger dynamic range of quantification. So we inject this virus into the mouse through retroorbital injection and allow the barcode to express. And then we use star map to measure the barcode sequence from the circular RNA together with the 1022 genes we mentioned during cell typing. By measuring these two information on the same slides, we can then derive the tropism in the cell type and tissue region resolvably. And here are examples of the spatial AAV expression heat map for individual slides. And interestingly, the brainstem actually, especially the thalamus and the hypothalamus, show the very high AAV expression compared to the cerebral cortex. We were able to quantify the PHP-EB transduction in individual mean cell types and the subtypes, identifying its tropism, preferring neuronal cell types and astrocytes. Epithelial cells are the least transduced ones. It is also worth noting that different subtypes within the main cell type, within the same main cell type, can vary a lot in their AAV transduction efficiency, as in the case of the astrocyte subtypes. So this emphasizes the importance of deep cell typing in the tropism profiling, and the mouse molecular atlas. A mouse brain molecular atlas really offers a valuable opportunity for this in situ deep characterization of viral tool tropisms. In summary, this work offers a comprehensive spatial molecular atlas across the entire mouse central nervous system at 200 nanometer resolution. We provided a careful annotation for over 1 million cells from 230 different subtypes, 106 molecular tissue regions, as well as the screen-wide AAV tropism chart for gene manipulation accessibility information. Importantly, it provides a roadmap for investigating brain-wide gene expression patterns and cell type diagrams in the context of brain anatomy. We were able to show the region specificity of individual cell types. We discovered new tissue architectures that complement established brain anatomy. And also, using spatial niche gene expression, we can better describe the function-related single-cell heterogeneity. The single-cell resolved spatial molecular maps provide unique analysis opportunities. For instance, the spatial patterns of cell-cell interactions and the spatial patterns of continuous molecular features. And furthermore, the analysis at the level of cell types and tissue domains with the molecular information can be a valuable anchor for comparative studies across species and conditions. We complement our data set with an online database. So it has an exploratory interface that can serve as open source for neurological studies across molecular, cellular, and tissue level. There's also raw data that can be downloaded from the single cell portal, and the computational code for generating the clusters and the figures are available from GitHub. I want to end by further outlook into other molecular tools we have in the lab. There is also study we published during the summertime called the RiboMap that can give the single cell result spatial translatomics information. So in this platform, we have additional probe that is annealed to the ribosomal RNA of the ribosome. And in this tri-probe design, only the RNA that's being bonded by the ribosome will give signal. So we're able to read out the RNA that's being translated. And there is a clear spatial pattern differences by comparing those ribosome bound RNA versus all the RNA, where the ribosomal bound RNA have this cytosolic preference in its spatial location. And furthermore, the StarMap platform can be applied in a context beyond just the nervous system. We also tried applying them in peripheral nervous system, but also other internal organs of the adult mice. And furthermore, I want to also point out that the efforts to build a consensus understanding of the brain cell types is a big research community efforts, exemplified by the Brain Initiative of the Cell Consensus Network, BICCN. So 2 years ago, there's special issue really focused on this multimodality network, multimodality characterization of the mouse primary motor cortex. And now the BICCN 2.0 are really extending their efforts to the whole mouse brain. And we are expecting to see very nice data sets and publication very soon. There are additional spatial transcriptomics tools like Murfish, Visium, StereoSeq, SlideSeq. They're all contributing valuable data sets on developing and aging brains. I want to end with a few more sentences on what our lab, Dr. Xiao Wang's lab, do at Broad Institute. We work active on tool development for understanding different flavor of molecules in a three-dimensional way in tissue setting to really look deeper into the RNA biology and the different aspects of the brain. Combining the molecular measurements with histology as in the spatial atlas, but also functional states such as electrophysiological signals. And with this, we want to utilize our understanding of RNA expression and regulation in the tissue to design better RNA therapeutics vectors for higher efficacy and specificity in gene expression in the gene therapy space. With that, I want to end with acknowledgement. As mentioned, this study is a really close collaboration between the Wang Lab and Liu Lab, and Dr. Yiming Zhou and the graduate student Yichun He contribute a lot to this study. We also got a lot of help from the Stanley Center at the Broad Institute, where the Brain Development Lab really helped us get the AAV part of the work set up. And also Dr. John Madison provide help with the neuronal culture that we use to optimize the viral construct we used later in the mouse brain AAV tropism mapping. And thank you for your attention. I'm happy to take any question, and I look forward to discussing more with everyone.
All right. Thank you, Hailin, for the wonderful talk. Please feel free to type your questions in the QA in the chat, or raise your hand for the questions.
Now there's one question.
Oh.
Yeah. Do you want to read the question? Actually,
you saw one? I don't see any.
Oh, okay.
I was able to see.
Oh, yeah.
the Q&A session.
Yes, from BG. Yeah, I'll read it then. Yeah, so initially you used a set of targeted genes about 1 to 11K for your method. How did you decide which genes or how many to use in your method?
Yeah, thanks for the question. So whenever you use a targeted approach, really the first big task is to determine what are the genes you want to include in this mapping. So let me go back to see if there's a useful slides for it. Okay, so one challenge in this image-based mapping is the crowdedness of molecules we may have in this image. It is important to resolve each individual puncta from the image to reliably call the mRNA identity. So currently, we can based on the dot density, it is important to avoid really highly expressed genes in your gene list to avoid this molecular crowdedness, so we don't have really say ribosomal protein coding genes or really high abundant housekeeping genes in the data set. That's one consideration. The other is depending on the purpose of your experiment. So in this case, we want to understand the cell type diversity. So what we focused on was marker genes that's already reported in single cell gene expression, a single cell RNA-seq data set. Plus, if there is more functionally related characterization purpose, say want to understand the immune response in a tissue injury setting, or want to understand cell-cell communication, then there will be more genes about the immune response, cytokine, or cell surface protein. So I think 1,000 gene is a nice number, which give you decent crowdedness of the dots, but also flexibility in spiking in the genes you want specifically for a certain experiment. Let's see for second part of. And yeah, with the single cell with the paired single cell RNA-seq data set, it is possible to extend its gene expression complexity. So right, so gene expression abundance and the experimental purpose are two important consideration for choosing the gene list.
I have one follow-up question. I think this was my question was, I was just wondering, like for 1,000 gene, around 1,122 genes for brain tissue you're actually working on right now, it works pretty well. I was just wondering, like for other tissues, so it depends, right? Which tissue you are working, and then how could we decide? You know, I think you already explained that, like we should avoid like housekeeping gene, like highly expressed genes, to pick up the genes for the sequencing. You know, is there any recommended numbers for that, or like 1,000 is basically the kind of around is best? Or should we actually just focus on those like marker genes, like well-known marker genes for cell types in your cell population? So could you talk a little bit more about that? I think you already talked about that, but I was just wondering if I'm studying a lot of tissues, and like how should I make a decision?
Yeah, that's a really good question. So we do have the capacity to extend to more genes by adding one more coding base in the identifier. So in the RiboMap study I talked at the end of the presentation, we have a probe gene panel for over 5,000 genes that we compiled from all the single cell RNA sequencing data of different mouse organs. So I think it is definitely doable to map more genes. And eventually for individual single cells, depending on its cell type, usually we have around 100, 200 different genes and about 500-ish reads, if you like, or the RNA spots in the cells. So it is definitely doable to have more genes to accommodate a specific need of experiment. If we really want to have housekeeping genes in this data set, it is possible to design to have them be read out in additional rounds of imaging. Say we have a combinatorial barcoding in the 6th round of imaging for 1,000 gene, and then you can have a say cycle 7 to specifically look at for other housekeeping genes with higher abundance, if you like, just to avoid decoding too many spots in the same imaging round. By sparsing them into multiple rounds, we can also achieve this purpose.
Hailing, I'd also like to follow up on that. For some, if you are going to image more genes for to avoid the molecular crowding, is your method compatible with expansion microscopy to make everything bigger so that they don't crowd together? I think yeah.
Yeah, it's doable to and it's compatible with the expansion microscopy.
Because you already put them in a gel, right?
Yes, it's already in the hydrogel. So by optimizing the monomer molecules we have, or by optimizing the buffering condition we have for the hydrogel, it is possible to do expansion microscopy. I do want to point out the size of the amplicons we have. If measured by electron microscopy, is around the size of 100 nanometer. So that's around the spatial resolution we could have for a confocal microscopy. So I do think expansion microscopy can help, especially say we want to look at say intron signal or splicing events happening in the dense packed nuclei. But otherwise, looking at the cytosine RNA is usually already okay without expansion. And also expansion comes with more time for the imaging as well. In the future, combining with spinning disk imaging, I think it's will be a nice combination of the time, cost, and also the RNA spot yield.
Oh, so far your imaging has been done on a conventional confocal microscope?
Yes, it's with like
takes a long time then.
There's no special instrument requirement as long as we have a nice confocal and say 4 color channels that can be clearly differentiated from each other, and then we are good to go.
We have one more question.
Yeah, so one more question from the chat. When identifying 230 different cell types in your annotation, did it involve manual intervention, or were you able to use an automated pipeline for this?
Right, this is a very nice question. It is indeed very challenging to annotate, to cluster and annotate these a million cells. It's a back and forth process. So we finalized with this 2-step annotation because one is we want to utilize the established nomenclature for the neuronal cell type. So we have this mean type level label transfer. And then for finer diversities, we cluster based on the spatial single cell data we have ourselves. So there is steps in filtering out very lowly expressed genes from the gene list for the sub-level cell typing. This is related to the challenging in the cell segmentation issue for the imaging-based spatial transcriptomics. So we do have to filter the gene list based on certain criteria to have better clustering results. The nice thing is that with the spatial information, we can always refer to what people already know about the distribution of a certain cell type in the brain sample. So we were able to confirm using the spatial information to know whether we have the right clustering parameters and the conditions. Yeah, so I want to say the cell typing itself is just based on single cell gene expression, and we have the spatial information as an orthogonal information for validation to validate our clustering performance. And I think all the code for this clustering process are available on GitHub. There is, and I think people can always download and reproduce the results.
I have also a follow-up question regarding the annotation, cell type annotation here. As based on what you told me, it seems like you use actually single cell RNA-seq data to have the annotation in spatial transcriptomics data. I was just wondering, like, do you actually also use like Weno markers genes to do the annotation also?
Oh, so in this integrated UMAP, we use the overlapping genes of the single cell RNA sequencing data and the gene panel list we have. So that's basically just the gene panel list we have to have this joint embedding and the label transfer. I think I should probably have backup slides for this.
Okay, so if they are clustered together, basically, for example, if they are clustered together, means that those cells actually are the same cell type. So you do the annotation
Right.
like
So,
in this way.
Yeah. So what we did is we first clustering this joint embedding, and for individual cluster, if the single cell RNA-seq label for this cluster is, say, the purity is over 90%, we think it's a successful integration and a clean cluster. In this case, we transfer the label of those blue cells to the red cells, the spatial cells we have in the same cluster. That's the criteria for cell label transfer. Otherwise, if the transfer is not successful, we then temporarily label them as NA cells, and then we later manually do this sub-level clustering by looking at what are the marker genes in those cells.
Oh, okay. So if we cannot transfer the annotation from the single cell RNA-seq to your spatial data, you will do that manually using the Weno marker genes also to check whether that Weno marker gene is expressed in your cluster.
Yeah, we'll later take care of them by looking into them one by one. Yeah, it does take a huge amount of efforts to do the annotation. But I think with the help of ChatGPT, we can have more smooth.
Okay. Use ChatGPT for type annotation.
It works nicely in terms of you give it a gene list, you can return the function of all those genes and potentially come up with a name, biologically meaningful name for that cluster. So it does help with accelerating the annotation purpose, but we do, but I think we only had access to ChatGPT at the end of our second round of revision. So yeah, it does help.
Is that just a default GPT or it's a custom chat that someone built?
It's just a default. We do this by mainly for confirming what we annotated. But in the future, I think there are active research efforts going on to use machine learning and this language model for annotating cell types. I think that's the pain in the neck for all those large scale.
Kind of like you feed in a gene list for geo analysis, and it gives you like terms that ChatGPT can guess the cell types in.
Yeah. That's interesting.
I didn't know that. I want to give it a try.
Yeah,
well,
I think
it is challenging to do the whole brain because not every like for example, I study the cerebral cortex. I don't know anything about the other brain regions. So any cell types in the other brain regions, I would love to have a resource to refer to understand them. Yeah,
so it's actually a very satisfactory process when we finally see all the cell types come together. And it's also like the case we see that histaminergic neuronal cell type at the first, so that marker is also positive for blood vessels. We once thought there might be some issue with the cell segmentation, but when we look into the literature, it's indeed a specific cell type in that specific brain region have that marker expression. So it's just a lot of fun to learn about the real cell types appearing here and there in the brain structures. So this one I was talking about. Yeah. Cool.
Great. Do we have any more question? All right. I don't think we have any more question. But thank you so much for the nice talk and great job, you know. And thank you, Ji, for hosting this seminar also.
Yeah,
you guys have a great day.
Organizing the event, and Hailing, it's a really great talk. Thank you so much. Learned a lot.
Thank you for having me here. Yeah.
All right.
Great. Good night. Bye.
Bye.
Bye.